Hi, everybody. Okay, the computer is talking to me something, but okay, about the recording. So today I will talk about, well, paleoceanography, but at the beginning I should point what is oceanography, because without this knowledge it is quite difficult to understand paleoceanography. Usually people understand that almost 3/4 of the Earth's surface is covered by ocean, but it is not really important information. The much more important information is that the seawater heat capacity is much higher than Earth heat capacity. It means that the most of the heat is stored in the ocean, and the ocean is steering the climate. I would like to show you, well, sorry, some extra information from a program covering the top of this map. I don't know if you see it. I just pointed to two towns on this map, Tromsø in northern Norway and Inuvik in the northern Canada. But can you see it?
Yes, yes. All is clear.
Okay. Okay, I can't, because there are a lot of information in the program and they are covering top of the map. But I use these two towns as an example how important is the ocean. In general, they are laying on the same latitude. It is a 64, little bit more than 64 degrees north. What means both of the towns are laying behind a polar cycle. What means that in the winter there is a polar night, and in the summer they have a polar day, 24 hours per day sunshine. And if you go to Tromsø in the winter, it is the picture from January, you will see a little bit snow. It will be cold in a windy day, like -5, but next day you can expect +5, and it is snowing and raining all the winter. And what is the most important? These fjords never freeze. What means the harbor is working all the year. I was also in Inuvik in January. For people who are familiar with the cold region, this picture shows everything. It shows cold, really cold. When I came to Inuvik, it was -64 Celsius degrees. And this smoke, it is not the smoke, it is frozen steam. So housewives, they are when they shop, they park a car at the front of a shop and never stop the engine, because to start the engine they need electricity from line. In America, it is 110 volts. It is really cold. And please believe me, I look like this next 3 months working in the Beaufort Sea, because day by day the temperature was more or less -50 to -60. And then in April, temperature rise to -35, and it was too warm for my clothes, so I went back to Poland. So why this difference are? We are in a very special position in Europe. So temperature in Europe is not according to the latitude, it is according to sea currents. So this red line shows North Atlantic drift. Our heater, it is the Mexican Bay. It is really huge bay. It is bigger than Mediterranean, Europe and Northern Sea all together. It is a huge bay surrounded by land. What means that water in Mexican Bay is very warm, and it evaporates. And the passat winds, the eastern winds, they are moving this humid air on the other side of America. So what means the evaporation is in the bay, but the rain fell down on the Pacific side. It creates very unstable situation in the ocean. So water in the Mexican Bay is very saline and very warm. This state of the ocean we call baroclinic. It is very complicated to explain it using the mathematical models, but it is possible to explain it very easy. But before I will explain it on the next slide, I would like to point these 4 yellow dots. So this one is very important for us. Laying in the Fram Strait. This is the strait between Svalbard and Greenland, the way to Arctic Ocean. Here, this water, very warm on the surface, flowing north, cools down and sinks. And this is the kind of engine. So watching this map, you see the global thermohaline water circulation. But for Europe, the most important part is this one. The engine causes that water start to circulate in the global ocean, also on the bottom of the ocean. On this slide, you can see the focus of the end of this current. So here is northern Norway, Svalbard archipelago, you know, probably the name Spitsbergen, the biggest island here, and then warm water going from Mexican Bay and then along the continental edge up north to Arctic Ocean. The red color, it is warmer water, and the temperature here is shown on a depth of 100 meters. And then you can see the arrows. They are calculated by very difficult model of baroclinic water circulation, but it is easy to understand baroclinic. Here you have a Mexican Bay and a very thick layer of saline and warm water. And then this layer is thinnering going northeast. So pycnocline, it is the gradient between less dense water below and high density, opposite, the high density water below and the less dense water on the top. So as long as the pycnocline is not parallel to the sea surface and the bottom, the gravity wants to move it. And then just the water on the surface is flowing this direction and the water below goes opposite direction. This is the baroclinic easy. And then it caused that the Europe get a lot of warm water around, and our climate is very, very warm. To measure it today, we use simple instrument. Well, simple. This one is relatively simple outside. It's a very strong pipe, but inside there is a computer and the memory and battery. And I can measure the 3 very important parameters by this instrument. Conductivity, which can be calculated to salinity, temperature, and the depth. All these parameters are measured in a very high accuracy, in this case, 4 digits after the point. And then you see Magda switching on this, Magda Łącka. She was a PhD student at this time. Today is almost professor in my department. Switching a machine by the magnet. It is waterproof. I can send this machine down in the ocean to 1,000 meters. You can see also winch here. We have a more accurate and bigger machines to measure these 3 parameters of the seawater. Here you can see 4 kilometers of a wire. This is very special kind of cable. We call it truss. So it is still rope outside, but inside there is a computer wire. The cable is very, very expensive and very heavy, almost 5 tons of the cable. It is funny because usually we use one watch to send the instrument 4 kilometers down under the boat, and the next watch to lift it up. One watch, it is 4 hours. To measure the ocean properties today, it is relatively easy. It takes time, but easy. You see the first result, a very simple result from fjords. The winter situation when the all water column is cold. Look, the temperature. The temperature is shown here on this scale. It is a minus 0.1. Sorry, it is minus 1.78. What means the water is almost freezing now. Also, salinity is very high. It's almost 35.4. And then dashed line shows you density of the water. This is very typical situation in the fjord in the winter. Okay, so one thing more, the density. Look, the units for density, 28. What does it mean? It means that in 1 ton of water, we have 28 kilograms of salt. Everything is calculated to the pressure of 1 atmosphere, just on the sea surface. And look, the summer situation. Summer, yeah, this is typical summer situation with the brackish water on the surface. So temperature is high on the surface, and then you can see a strong gradient of temperature. We call it thermocline. And the salinity, it is another continuous line, also halocline here, what means the surface water is not mixing with the water below because gradient of density, pycnocline. So this is very easy in case of today measurements, but how to study the ocean property in the past. So most of this information are recorded in the sediment. So most easy instrument and sampler for sediment sampling is Van Veen graph, but not very useful in the paleoceanography because it gives us sample which is disturbed. So we use a gravity corer. This one is very, very simple, prepared by myself for modern foraminifera study. It's very easy to use, very fast, not for great depth. We use it maximum to 300 meters, but the results are perfect because we can very easy to slide the sediment, which is in the plastic pipe. This plastic pipe is inside the metal one. It is transparent. We use a piston from the bottom, and then we can cut 1 centimeter slice very easy and put them into the jar. And here you can see the alcohol samples are preserved in pure alcohol with Bengal rose, just to stain living foraminifera. So this knowledge about modern foraminifera biology is extremely important just to understand what our proxy are showing us. Here you can see more serious instrument. These 4 girls really know how to use it. Believe me, the professor on this image is only to make this image nicer. These girls are using this box core, very heavy and complicated machine. They call it Loki. If you know this Nordic god, it is the kind of very bad joker. It jokes sometimes, you know, after half an hour of moving it down and next half an hour up, and then it is empty. It's not nice joke. But if everything is okay, you can see how this machine is working. More or less 50% of oceanographers believe that this machine gives us undisturbed sediment core. I'm included to this part. So when this instrument is landing on the bottom, lead placed here, move the box and insert the box into the sediment. And before it is lifted up, this arm moves the spoon down under the box, protecting the sediment inside. And then after 1 or sometimes 2 hours, we have a sample on the deck, and you see Joanna Pawłowska. You could hear her last week. She's sampling sediment from a sea bottom. this is just a piece of sea bottom on the deck of Oceania. From, well, not very great depth, I can see the landscape around is Hornsund probably. So the depth here is about 200 meters, but we use this machine even to 1,500 meters without any problems. And next and last machine I want to show you, it is the gravity corer, the most serious machine. It looks beautiful when it is hanging on a crane, and if the weather is such nice like here in Hornsund, but it creates a lot of problems on the open sea when the boat is rolling 30 degrees one side and 30 other side. To keep 1 ton of lead on the top is not really easy. And the whole corer is 4.5 meters long in 3 sections. And then you can see the area where we used to work. And it's a lot of work to receive samples like this, small, tiny forams. Putting together all pieces of this machine usually takes us about 2 or 3 hours of work on the deck. You can see that our deck is not enough long to put it, so we have to use many tricks to put together the steel pipes, and then we have to place the inner pipes standing somewhere at the front of boat. But finally, when the sampling is successful, we have sediment core in 3 sections, 1.5 meters long, good protected, and then we store them in a refrigerator in 4 degrees and no light. Here you can see the core catcher. It is very important part of this machine. This is a kind of springs protecting sediment inside when we move the core up. And after sometimes few months later, we are in the lab. You see the sediment section of the sediment core in the lab, and this is the most interesting part of science, to open the sediment core and start to study. Everything what I showed you before, it is the field work. It is of course funny, but it is just the field work. Science starts in the lab, and then opening such sediment core, it is the big project which we plan sometimes discussing even few weeks. What kind of analysis we want to do, the method for opening, and well, the first step, we do magnetic susceptibility. It is very useful method just to check amount of ferromagnetics inside. In fact, the iron is connected to the sun. So if susceptibility is high, if it is high, we can expect that there is more sand inside. So this one we can do without opening, just in the lab. And then sometimes we have such results which surprised us. We got this result more than 1 year ago, and today we know what is this. It is the result of Storegga tsunami, which occurred in Europe 8.2 thousand years ago. So you see that these first measurements we can do without opening sometimes are very, very important. Another thing which we could do is without opening, it is X-ray, in this case, computed tomography. We used to do in the hospital. To get such high quality picture like this, the image like this, we have to do something like 5,000 shots. What means that radiation, X-ray radiation, kills the man immediately. It kills the DNA also. So we stopped to do this kind of images. However, they are very, very useful because you can see all hard things. In this case, there are drop stones in the sediment. You can see also the shells in the sediment. So we can organize the cutting according to the hard elements inside. But of course, sometimes it is possible to do such shots, such X-ray images without killing the DNA in both half of the sediment. 2 or 3 years ago, we had the possibility to open the core in the Bergen, in Norse laboratory. They have also this DNA laboratory, very clean. And then after opening, we took one half by hand and we went to another building, the University of Bergen, and they help us to make these pictures. And you see that these pictures are really useful. You can see some demiction on the top. They were transported by iceberg. You can see nice lamination here, which after about 2000, almost about 2000 years ago in this place. And also you see lamination here with the many drop stones during melting, beginning of a Holocene. So X-ray images are very, very useful. However, dangerous for metabarcoding analysis for any DNA, ancient DNA study in the sediment. So we have to cut the sediment first. We use this vibrative saw now. In the past, we were using Flexa, but the Flexa dirty lab. But using this machine, we can open it without big problems. We can cut the plastic, and we do not open the sediment in this lab. So after cutting plastic, we clean everything, and the core is transported to super clean lab for Joanna's study. And she and her team, they open this core to part. One part is stored in their lab, and the other part is going back to our one, where we start, well, first description of the sediment. It's just very important. You can see the color scale, the Mansell scale, defining the color of the sediment, and every information you can see on the surface are very, very useful later during interpretation of the data. So after description, we start to use X-ray fluorescence. It is perfect method, especially the instrument we use. This is Olympus Vanta, but there are many other instruments for this. It is very fast. So in 1-2 hours, we have a full information about chemical elements on the sediment surface. So you can see that the sediment is protected by very thin polyethylene. It doesn't impact the measurements. And usually this machine, it is the hand machine, but it is not nice to carry it 1 or 2 hours. So we build a special frame for this, and then we can do measurements very easy. On the picture, the Danushka Devendra, my PhD student from Sri Lanka, he's really master of this kind of modern method. And look the results of this one. I will not talk about everything, but let's say iron to calcium, which is perfect to understand what happened in time. You can see, well, this is just an aspect ratio. So when the values are high, it means the income from the land is high, because iron is coming with the suspension, with the terrogenic matter, the mineral terrogenic matter, and calcium is connected to carbon carbonate. This one is produced in the water column by the organism. So you can see how it was changing during the Holocene in Hornsund. More or less 8.2, you can see a significant change in the sediment, decrease of matter from land, and then during the rest of Holocene, it is increasing one more time. What means the glaciers stop to melt here, and then they melt one more time and release a lot of meltwater into the fjord. Titan to calcium is more or less the same, and beryllium to Titan is like a mirror, because Titan is it's just the aspect ratio is in the other side, but beryllium is connected with the chlorophyll, which is produced in water column by diatoms and dinoflagellates. So you can see that the simple machine, the X-ray fluorescence, help us a lot to understand what our sediment core is showing. And the next part of my show is about more advanced proxy, but maybe I would like to make a short break. I have to drink a little bit. Is it possible? Marek, can you hear me?
Yes, you prefer to have like a few minutes, or it's just...
Let's take 10 minutes break.
Okay, so let's meet in 10 minutes. So then we will extend a little bit, but as we discussed before, that is possible. Okay, so let's meet 10:40, right? Okay.
Yeah, 10:40. Yeah. Pollen's in there. I have an allergy for this. So may I start now?
Yes, it looks like we are all ready. Yes, you can go on.
Okay,
please.
Thank you very much. The very important proxies we can get from foraminifera. In the Arctic, they are very common in the sediment, so we use them as a main source of information. The stable isotopes, it is perfect method to study the two very important parameters I showed you at the beginning, temperature and salinity. And the oxygen is perfect for this. We have two stable isotopes of this element, 16th, which is common, and 18th, which is rare, but still our machines, the AMS, the mass spectrometers can detect it. So the aspect ratio between these two elements shows us temperature. How it works. During the glaciation, the large part of the fresh water which evaporates from the ocean is stored on the land. Since the stable isotopes are used like this, that the light one is more mobile. So the vapor water mount more the light isotopes, and the heavier one stays in the ocean. So the process during the glaciation is that the ocean is heavier. It is not really correct to say it is heavier, but it is commonly used in a common language. But don't write it in your articles. So in the articles we point that the delta is increasing or decreasing. But in the such talk I will use the heaviering and lightering which is, if you don't believe me that it is not correct, try to translate it to Polish. It is impossible, in fact. But anyway, during the glaciation, what means when climate is colder, the ocean increased the isotopical weight and increased the salinity, because a part of the water which was in the ocean is stored on the land. So there are two very important information. So if we understand the process that the light isotopes go to the land and they are removed from the ocean, so we can calculate the temperature. Oh, sorry, but I should say something about the units. I forgot about it. So we use two units, the PdB and SMOW. The second unit, the standard mean ocean water, is used in environmental science, but in paleontology, in paleoceanography, we use a PdB units. It is unit created on the basement of PD, belemnites, Cretaceous belemnites, but this unit is very useful and only used in paleoceanography. So so we can use this two isotopes in this model. You see on the top of the slide, I can't to see it, but the model is terrible. It's very long, and then, however, it is useful, but useful only on the open sea, the really open ocean. When you are close to shelf, it makes a lot of troubles. And look, the example, I just calculate the temperature using the data from the core taken in the Storfjorden. It is the western part of Barents Sea. Very close to this place, there is an oceanic front. What means on this side, we can see Atlantic water. On the eastern side, Arctic water or Barents Sea water, which is less saline and colder. This water is more saline and warmer. And as long as nothing happened with the oceanic front, model shown on the top works. But I calculate the temperature for this core, and look what happened. We have here the temperature, which is independent of stable isotopes, or the temperature from Greenland ice core. Just after Younger Dryas, both of this data shows warming, because it was warming, but our data shows something opposite, cooling, and very significant cooling. How it is possible? Well, it is not a cooling. This is the water mass change. The oceanic front move east over our sampling station, and it shows the change in seawater properties. It is not the temperature change. The same in the middle of ocean. It is warm, stable warm, and then we can see cooling. And in neoglacial, we have a cooling on the surface and something like warming on the bottom. Of course, it is not true. All these changes we can see on this graph are connected to the water masses change. What means the position of the oceanic front was changing over or moving over our sampling station. So this model is good, but not in 100%. However, it is possible in this case to calculate salinity. Because if we have a temperature from an independent source like alkanons, I will talk later about this method. So using this model, after transformation, we could calculate the salinity of the seawater. And in some points, it is possible, of course. Look, this layer I just point by red arrows. in this place, this model is working, but in other place, not necessary, because, well, the temperature from alkanons shows temperature on sea surface, and the salinity calculated from stable isotopes is from benthic forams. What means it is benthic salinity, just near the bottom. So I just calculated, I prepared this transformation, but I never published it because it is not really useful. The useful is just to show data of oxygen delta, but remember that we have to show it in the opposite direction. So this scale is always inverted. Why? Because when the curve is turning to the right, it shows warming, and to the left, cooling. And this is standard method today in the paleoceanography. We do not calculate the temperature, we just show the results of our stable isotopes analyzed. You can see the carbon shown exactly the same, just on the scale. And now, if you watch, it is not the same chord. This one is from mouth of the Hornsund. You can see that all the Holocene we have increasing the temperature. Then beginning of neoglacial is cooling, and then temperature varied. And here you can see last little ice age, which is quite cold, and very characteristic variation of data during the melting. So this is very typical because still the large part of ice cap is on the land, and during the warming, this ice cap removed a lot of melt water into the seawater. So we have this variation, cooling, and then meltwater release, which is in fact seen as a cooling. Next stable isotope we use is carbon. Carbon is perfect, but very difficult to understand because its distribution in the Earth is complicated. So general rules are like this, that the ocean is heavier than the land because lighter isotope is lighter, what means more mobile. So it is easy to transfer the light isotope to the atmosphere as a CO2, and it is used by plants on the land. So land is always lighter than the ocean is. But if we see the detail in the ocean, so surface of the ocean is lighter than deeper part. Why? Because most of biological processes occur in the surface, in the euphotic zone. So surface is lighter than deeper part, than abyss. But if we go to shelf, the shelf is lighter than open ocean because there are more biological processes on the shelf, and then that we have some stable isotopes coming with the organic matter from the land. So you see that the distribution of the stable isotopes is quite complicated, and everything depends how fast are the biological process. It is very good. Very good to use an example from a mouth of the river. In this case, it is Vistula, just at the front of Vistula River. You can see the sampling was in a different season, and the most important season is in March. The March is typical spring in the northern part of the world. The biological process starts after the winter. Winter is dark, so primary production is almost disappearing in the winter because the sun is very low over horizon, and then due to albedo, the transmission of the light into the water is very low. But in March, the sun rises enough high, and then it starts the processes. Water is mixed, what means the nutrients are removed from the bottom up to the water column, and the diatoms start to be active. And then look, the value of carbon data, they are very, very low, what means we have more light isotopes in the water column. And later on, late summer, also autumn and winter, they are heavy. Just simple mechanism at the front of Vistula River. But stable isotopes were used first time by Hoffman to explain very catastrophic glaciation, the snowball. I like this example because Paul Hoffman made first time he made a really good job using this stable isotopes of carbon. He made it in 98. It was many years ago, and he was probably, I think, one of the first who used stable isotope as evidence of glaciation, very old glaciation, almost billions years ago. So, in fact, the snowball was first time pointed by Budiko, this hero, Russian geologist. In fact, for me, he is a kind of hero. Look the date when he published this data. It was the communism in Russia in that time, and the tectonic and genetic was forbidden in Russia. And despite this political problem, he published the data regarding the supercontinent which was formed, and he named it Rodina. This name was created by Budiko. Kirschwing, he just described how the Earth melt after this glaciation, and he used a very nice name, the snowball. So the journalist picked up this name, and then he was very known after this publication. But Paul Hoffman made excellent evidence of this glaciation using these stable isotopes in stromatolite phase in Namibia, in Africa. So during this time, the supercontinent break down and split to many different plates, what means that the length of the shelf increased so much, and the stromatolites made a kind of monoculture on the Earth. They start to produce a lot of carbonate as a cover of their colony, and they produce oxygen. So this is why we have oxygen in air, in atmosphere, and they use the carbon to build their body. And the process was so strong that Hoffman described huge depletion of the carbon delta, even -6 per mile. So it is a lot. Here you can see the stromatolites. I shot this picture on Dunajan. It is very small. There are very small islands in the mouth of Hornsund Fjord, and then you can see very characteristic arcs. These arcs, it is the cover of the colony. Stromatolites, I think they are still in many places in the world, in California Bay, as I remember. But anyway, these tiny cyanobacteria changed the world in that time. And today, we can use a stable isotope just to make evidence of their activity 900 million years ago. Today, I use stable isotopes as a standard method in paleoceanography, and I can show you the example from a core taken from eastern Greenland continental slope. So this core was taken on this plateau, which is on a depth of 1,200 meters. Here you can see the East Greenland Ridge, and this is the shelf which is shallow, about 300 meters only. So this place is 1,200 meters. The aim of this study was different, just to show the return Atlantic current. So the current, which is this warm water, Atlantic water coming to the Arctic, it is flowing along the continental break and slope, and part of this water reach Arctic Ocean today because of warming, but it was not every time in the past. Part of this water turns to the west and goes back along the Greenland continental shelf, forming beginning of the Greenland Gyre here. Greenland Gyre, it's the gyres in the ocean are not like on the river. So in the river we can see this very fast moving water. Gyre in the ocean, it is huge mass of water slowly circulate in the sea. So the aim of the study was this RAC, we call it, Return Atlantic Current. But I'm not going to talk about everything because it will take a lot of time, but I want to point only one place. Look, the carbon delta in foraminifera, benthic foraminifera. And in this core, 20,000 years ago, this is the last glacial maximum, we have a lightering, just a lightering. How it is possible? Very cold period, and what we can expect during this time, okay, I will go back for a while. During this time, we can expect that the ice cap of Greenland, the edge of this ice cap was somewhere here, very close to sampling place, and we have a lightering. Lightering, what means that on the bottom, a lot of fresh organic matter upper. Something strange, because usually in such situation we can expect old matter which is heavier, just heavier. But I show you this example just to point very important information. The one proxy is usually not enough to understand sea environment. This is why we use a number of different proxy, and trying to explain this result, we have to see everything what is shown on this picture. The first is sediment accumulation, which was very low because the glacier advance. During the advance, the melting is slow. The melting is fast during glacial retreat, as you can see here. Unless late glacial or, for example, Bolling, Alrod, they are very warm 2 periods. So during the melting, we have a high sediment income. And 20,000 years ago, it was glacier advance, low sediment income. And look, the foraminifera. I can't to see the top. Okay, so up here I can see. So this is the total foraminifera flux, very low. So very rare foraminifera, benthic foraminifera, receive very fresh organic matter from the surface, and the seawater was relatively transparent. I mean, no turbid water, because a low amount of sediment was coming to the bottom. It created a story. The story was like this, in my opinion. Close to glacier front, there was a very cold water which was frozen. I mean, the pack ice was covering the sea at this time. Under the pack ice, ice-connected diatoms are blooming at the spring, and there are no consumers for this organic matter produced by diatoms. So the export of organic matter is very high during this process. So this is why we have a lightering of organic matter on the bottom here. However, our benthic foraminifera flux is very low because this income of organic matter was once a year. So just at the spring, and the rest of the summer, it was the muddy water, or sea was covered by the ocean, by the sea ice, and the organic production was very low. It was just the glacial, maximum of glacial. However, the result of benthic delta carbon is showing lightering. It looks like high productivity ocean, but it is not true. The productivity was only for a while, and today we can observe it sometimes in the fjords. As long as they are covered by ice, we have a very strong spring bloom of phytoplankton, but just for a while. And during this bloom, the export of the matter is very high, down to the bottom, sometimes such high that the bottom of the fjord is covered by green mats, but just for a while. And next step is the melting of the glaciers, and the organic mineral matter transported from a glacier is covering this green mats. The result of this is lamination in the sediment. We have these black layers and the lighter mineral layers over the black layers. So this is the example how to use stable isotopes and how to understand the results. It is quite complicated. We need a lot of proxy to understand the processes which occur in the past in the ocean. And on the end, I would like to show you two, two proxy, which I know this slide is not nice, but I show it only to point this publication. So most of this information you can get in the article we prepared some years ago. Well, in 2015, all these methods were new. Today, they are standard in our lab. However, not these analyses are quite rare in Europe. So only a few research centers are doing such analysis like IP25 and alkanons. They are complicated. We need chemists, really professional chemists, to do this analysis. We are happy in our department because we have a chemical lab. They called marine pollution, but 2 or 3 very good quality chemists are doing this analysis for us. We use for this gas spectrometry, complicated. I never touched this machine. Only professionalists can use it. Alkanons. I will not explain you what does it mean. What means the details in the biology of Coccolithophora, but in general, they produce alkanons as a biological compound, and these alkanons are different depending on the temperature. They form the chains in which a different number of carbon atoms are fixed. So in cold region, more the carbon atoms are fixed. And then using these complicated models, we can calculate the temperature and temperature on the surface. And this is the mean year temperature of the surface. It's perfect information for paleoceanographers. Another proxy is IP25. This bioindicator is produced by diatoms, but the diatoms connected to the ice, so sea ice. So using IP25, we can detect when the ice was common in the past and when it was rare. And this is example of this study. I like to show this example because we spent many years together working with the Magda Łącka, many years to get this result. The first step was the publication of full data from a core taken in Storfjorden, Renna. The Renna is an underwater canyon formed by glacier, which first glacier made this fjord. Today it is not a fjord because it is open here, so it is straight, but still we use this name Storfjorden, and this deep canyon, underwater canyon, is a perfect place to study oceanographic change in this region, because we have Atlantic water here and Arctic water on the eastern side, and a month of Atlantic water coming to the Arctic. It is the parameter which steers the climate of this region. So still I can't to see detail. Okay, I will move. Sorry, I will move one window here. On the graph, you can see here, you see the black line. It is our data, alkanons, showing the temperature on the sea surface. Look, in the younger Dryas, this orange or maybe red, or I don't know exactly, not black line. It is the temperature which was calculated by Mimi Oxman, the Finnish colleague. She was preparing her doctoral thesis basing on data from Disco Bay and Baffin Bay on the other side of the Greenland. And look what happened. So we have this period when the temperature were varying very much in the Bolling and Hanrod, and then cooling at the beginning of younger Dryas, and then alkanons disappeared. And then if we go to foraminifera, this is the flags of foraminifera, forams also disappeared in that time. Strange. It looks like catastrophic. But on the other side of Greenland, it is warming at the beginning of a younger Dryas. The Mimi Oxman made excellent work. She calculated this temperature on analyzing diatoms. She is diatom specialist, and then it could be really difficult to understand what this data are showing. But thanks to very good eye of Magda, she was just watching the sediment, and in one moment she could see the very little blue flash in the sediment. And then at the beginning we thought, this is nanodiamonds. You know, the one of the theory why there was this younger Dryas catastrophe was the comet. I don't like this kind of explanation, because it is the easiest, and usually we have no any evidence of this. And then we found the nanodiamonds. No, terrible. But after detailed analysis, we made it in the Poznan in the upper that it is vivianite, not the diamonds. Vivianite, it is also very strange, because the vivianite can be formed in an anoxic environment. So vivianite is common in the lakes. It was only one observed in one of the Siberian river mouth. They used to freeze in the winter, so anoxic environment can be possible in the mouth of such river. But how is possible anoxic in the middle of ocean? It is only one explanation for this. All these are frozen, but it was covered by perennial ice, not the sea ice. Sea ice is broken. You know, the sea salt ice is very soft, so gas exchange between frozen Arctic Sea and the atmosphere is possible. But when the big area is covered by freshwater ice, well, it restricts gas exchange. between sea and the atmosphere, and then it is great finding, because then we start to study the details and we create a okay, sorry, but this is the Alkanos. But look, the IP25, another proxy showing the ice. We have a continuous ice cover during the Younger Dryas. So altogether, if we put this data, we can create a theory about the Younger Dryas was steered by oceanography and the melting ice caps, but both ice caps. This one in Europe, here you can see it, and this one in America and Greenland altogether. On the onset of Younger Dryas, Bering Strait was still land. Only the way for fresh water coming to Arctic Ocean was the Fram Strait and the Barents Sea. So on the onset of Younger Dryas, we had a huge mass of fresh water flowing down south to Iceland, Great Britain, and then even to Atlantic Ocean. It blocked it blocked the warm and saline water coming from Mexican Bay to the Europe. What means our heater in Europe was stopped. But part of this water turned to the north and went directly to the Greenland, to Disco Bay, to Baffin Bay, to Labrador Sea, warming this part of Greenland. And many fjords in southern Greenland just were deglaciated. Glaciers disappeared during this short time of Younger Dryas. So oceanography of this region was totally changed during beginning of Younger Dryas by the fresh water coming from ice cap to the Arctic Ocean. The cold was only, I will go back to the previous slide, the cold was only the beginning. And then you see another pulse of colder environment, and the third pulse, which is in fact preboreal oscillation. So warming during the Alrod and Bolling and Alrod caused that ice caps melt, and the meltwater cooled down the Europe so much that okay, we are at the beginning. I'm very fast, but I haven't used to talk so much. We are at the beginning, but then we can conclude that the ocean steered everything, also our culture, because look what happened at the beginning of Younger Dryas. North Atlantic drift was discontinued in Europe. So this water turned to the north, warming Western Greenland. And the Europe in that time was very cold, such cold that this fantastic culture of Cro-Magnon people disappeared. These people died because Europe was cold and dry in that time. And nothing worth is cold and dry. Cold and wet is no problem. You know, the Little Ice Age, for example, was cold, but it was also wet, the huge income of snow. So vegetation in the summer was perfect. We could alive. But the Younger Dryas, it was catastrophe in Europe. It was also catastrophe for people who were living here in Anatolia. Natufian tribes, for example. So everything what they could do is just to escape south to Mesopotamia, and they started Neolith. But the beginning of this process was the catastrophe in Europe, but only in Europe. So 10 or 15 years ago, the people thought that the Younger Dryas, it is the general cooling. No, it was the regional cooling in Europe and the European Arctic. Everything stays ocean. And then why the Paleoceanography is so important. So thank you very much for your attention. And if you have a if you have a question, very welcome.
Thank you so much. Thank you so much, Marek. Yes, yes. Your questions are welcome as well. So I hope that PhD students are still around. So yeah, we see Carolina and Carolina, and we don't see Harry, but Harry is around. So yes. So we can see your faces, which is great. Hello, Eman, Tomasz. Good to see you all, Agnieszka, and all others. We have many guests from mostly from the Institute of Oceanology. So anybody can now ask questions. So you don't have to be a PhD student, but of course your questions from students are most welcome. Impressed or surprised, or you know, something is weird, maybe not so clear. This is time to explain. Yeah, again, I see that Stefano is not attached. That means that we don't have our most active student, but maybe Tomasz or somebody, yeah. Still quiet? Yeah, I actually may have maybe the first question. So if you compare that onset of youngest dryas, could it be that we may observe something like this, and we may predict, or you may predict something like this in nearest future, in 50 years or 100 years, that something like this might happen soon, so that Gulf Stream will be less strong or not strong enough to warm up Europe. So with the global warming, we will get European cooling, which is kind of predicted. But what do you think about it? Is it likely, or it may not happen because this is another situation? That's actually quite exciting, I would say.
It is widely discussed now because we can see this problem. We have still ice cap, the Greenland ice cap, and it is melting so fast. So we know that it is increasing sea level, but in my opinion, the situation similar to beginning of younger dryas is impossible today, because there is no another ice cap, the European one. The Bering Strait is open. Still, the canals between Greenland and Ellesmere Island, all these canals in Northern America are open. So the fresh water coming to Arctic Ocean have many places to spread down south. The younger dryas problem was that there was only one way for this water. It was from Strait and the Barents Sea. And then also next problem was amount of this fresh water, meltwater. It was much higher than today Greenland can produce. So I don't think that it is possible to repeat younger dryas. However, meltwater coming from Greenland can also regulate the North Atlantic drifts.
Yeah, and that may mean for us that Europe will be getting colder, but the global warming will continue.
Yeah,
which might be on one thing may be good because Europe would be colder, but I wouldn't be happy. Well, I don't think I can observe it. I hope I won't observe it, but anyway, it having very cold and dry Europe, that is something which is maybe a bit scary.
It is catastrophic. Now, the dry
Sounds
and
like.
cold, it is catastrophic. Please believe me, the cold and wet is not really problem. Look, the little ice age in Europe, the productivity in Europe was very high. The Poland in that time was very rich. We export a lot of food to the rest of Europe during this time. So cold and wet, it doesn't create really problems. So of course it was before we made amelioration. So the snow cover in the winter was excellent, because at the spring we had at this time we had a lot of swamps in the Poland. They catched a huge mass of water in the environment, and this water was enough for a hot summer. So today we created problems because we don't have a system to catch this water, and the most of the water goes to the Baltic Sea. But the cold and dry, it will be catastrophic. The production in Europe, I mean, biological production, will decrease immediately.
Right, right. Well, so it looks like then our last chance is to migrate to Egypt, Eman. Yeah, maybe Egypt would be like optimal, like more wet, and but it will be maybe hotter, so much hotter. So no, maybe not.
No, it's much better to react. I mean, to do something in Europe, well, we have to be prepared for a future. So today we know that we have to change this water system in Poland. We have to do everything what is possible. So as a normal people, for example, me, I catch about 6 tons water during the rain in my tanks. But this is very, very little things I can do. But the state authority, they can do much more. So we can catch the water in the swamps. We can rebuild the swamps. It is not a problem. It's just a decision, nothing more.
I guess we have specialists on these things, yeah, from the Institute of Geography in general from Warsaw. So if you can share your point of view, yeah, is it possible to rebuild swamps in Europe, in US, in I don't know, everywhere, or it's like sounds like a dream or impossible at all?
It is not so easy. The best example is a discussion about the new EU regulation. I'm not sure how Zielony Ład, how it is translated to English.
Green Deal.
Harry would.
Green Deal. I think it's Green Deal.
Green Deal. Yeah. Okay.
Green Deal. Okay. Sounds like deal. Yes.
So this is our voice. The scientists create this Green Deal. This is our opinion. And look what happened. Everybody are against.
Nearly everybody's against. Yes,
yes. You know, they don't believe us. They believe farmers. The farmers are nice because they want to use all these chemical stuffs. They want to use tons of fuel to produce food, and they don't want to change anything. Nobody is listening to us.
Yeah, this is partly our fault as well. I mean, this is the fault of education. So I mean, first should education come and explain, and you know, we have a big gap because these things are coming, but people don't understand it. And of course, there is, you know, reaction, politics, and whatever. So then it's like, this is our, actually, this is the role for younger generation of scientists and teachers, lecturers to actually, but it's maybe kind of late, but it's never too late. So So you are the future, guys. We all have to do it, but especially you have to do it.
Yeah, definitively. Everything depends on the young generation, because I think we have no power to change it. So young people are needed here.
Right, right. Yeah, I am more than sure, but we don't see Yannick, but usually Yannick has some questions, and I'm sure that at least one question. Yannick, would it be possible to kind of?
Well, no,
No?
today I have no question. I just have like note for myself that I need to read a lot of how this carbon delta proxy works, because well, I kind of get it, but now I need to renew this knowledge. But this is something that I need to repeat. So it's no
Yeah,
for
it
me.
is a very useful proxy oxygen isotopes as well, and many others. And that is actually the main take-home message, I think, Marek, what you actually express a couple of times, that paleoceanography, all these reconstructions we do have to be based on multi-proxies. Yeah,
Yeah.
so this is the only option.
Yeah. It's, and also, what is very important, we have to create a new proxy every time, So this is the excellent example is my department, and we invite a laboratory of marine pollutions. Małgorzata Szymczak-Żyła, she's head of this lab, and 3 good chemists, high quality specialists in chemical analyze. They are really great in paleoceanography, so they can do something what we have no chance to use these machines. We don't know how to use gas spectrometers and all these details. How to clean the samples. They use a lot of tricks to clean sediment, to separate some chemical compounds. So we have no experience. We know the theory, but they know how to use these analyzers in the practice. So combination of chemists and paleontologists, it is the perfect idea.
Right. And a chance to, yeah.
But by the way, so this is our discover. Just a few months ago, Małgorzata, the head of this lab, came to me and she said that she has an idea how to measure compesterol in a sediment. Compesterol, it is like a lignin. It is produced by plants on the land, only terrestrial plants. And then this compesterol is very good preserved in the sea sediment. And we made the first analysis. And then it appeared that we have another proxy. Just, we can check how the land income, I mean, this organic matter from the land was transported to the in this case, Hornsund Fjord. And we can see the increase of land productivity, terrestrial productivity in the sea sediment. This is perfect. Just accident, a short talk with the chemist, and we have a new proxy. So this kind of cooperation is really needed in the science, in paleoceanography especially.
So now we all know why you are invited. Yeah, so that is that we have to search different approaches, different tracks, and collaboration, yeah, between different disciplines. But with this compesterol, would it work well in all climates, so in warm seas? So I mean, this lignin is pretty well preserved and transported And I mean, with all temperatures, all climates, or it's more like Arctic thing, subarctic?
I don't know how it is preserved in a warmer climate. We haven't checked it, but in the Arctic, it is very good preserved. But also in the Baltic Sea, we check it in the Baltic sediment from Gdańsk Bay, and we can detect compesterol and beta sterols in the Baltic Sea sediment. So we have a new proxy now.
Congratulations, very good.
Yeah, but this is not for me, just Małgorzata, Professor Szymczak, she's a hero in this case. I'm an old professor, just almost retired, and I just organize the work of your young people, but they are really good specialists. And this is, as I showed you, that the 4 hero girls can use very big machine on the deck of Oceania without any problem, and then they analyze samples, and they are future of science. Not me. I'm old man.
Well, not that old, but I was so impressed with all these photographs, and I'm very jealous as well. If you need some help, I'm happy to join you. But I know that it's not so easy. I think you are very enthusiastic about all your stuff. Is very happy to join all these cruises. That's my guess. Yeah, or do you have problems with stuff?
Yeah, I need help. Yeah, so that's problem. That is true, and a lot of help. But not only in the field, but also in the lab. So I have a lot of new ideas what to do, but the number of people is limited. So
okay, this is the absolute last chance to ask the last question or a comment or anything else. If not, yeah, don't switch off your computers. We will continue with some let's say logistics and organization of your oral presentations. But Marek, thank you so much for the great talk and sharing your knowledge. So I hope you will come back maybe next year, or if we organize something like this, but you are always welcome. So